We continue, and it is my honor to introduce first our moderator of the last session of today's Evolution Club lecture series event. Our moderator is Christoph Winkler, who is an associate professor and deputy head at the Department of Biological Sciences at the National University of Singapore. Christoph studied biology in Munich, Germany, and did his diploma thesis at the Max Planck Institute for Biochemistry in Munich. He obtained his PhD from the University of Würzburg, Germany in 1994. In 1996, he became a Human Frontier Science Program Fellow and was later on a postdoc at the Howard Hughes Medical Institute at the University of Washington in Seattle. He returned to Würzburg in 1998. Then he made his habilitation in biology in 2005, and then 2007 he joined the National University of Singapore. I don't want to continue further. Christoph, the word is yours, and you will be the chair of the remaining session. There are microphones, both mobile, yes, and also some fixed. So, Christoph, the word is yours.
All right. So, welcome back, everybody. It's actually my pleasure to be the moderator of this session. I think we heard two fantastic talks, absolutely great science. The floor is open for questions. Again, I would like to ask you to go to one of the fixed microphones or raise your hand, and then we will pass you a microphone. And if you could identify yourself, that would be awesome. Do we have questions?
Yeah, Barry Halliwell from NUS and A-Star. You mentioned melanin in the eye tended to stay around for a long time, and I wonder if that's true of melanin generally and why it is.
Yes, it is true. I should say that the study of soft tissue preservation in vertebrates is moving ahead really rapidly at the moment. Of course, it's been known for some time that it occurs, but there's a lot more focus on it now than there used to be. There are technical advances now with the application of very sort of laser geochemical styles of analysis to actually get an idea of the composition of the material. And it was a bit of a breakthrough a few years ago with the recognition that sort of ovoid or egg-shaped structures which have been seen under the electron microscope frequently in these sort of black areas, which had been thought to be bacterial preservation, are in fact melanosomes. It now turns out that in the retina, in feathers, or generally in the skin, if melanin is present, it tends to preserve extremely well, and that in fact it's often really what gives you skin preservation at all. If you think, for example, about those rather famous Mesozoic ichthyosaurs from localities in Germany, this sort of in shape rather dolphin-like reptiles, where you can see the dorsal fin and the tail outlined. The only reason why they preserve is that the skin was heavily melanized, it seems. I guess it's basically something to do with the character of the protein as such. It cross-links readily. It's rather antibacterial in character. I mean, it's part of the immune system of various invertebrates and so on. And so it just seems to be very resistant to breakdown and, of course, show up with a nice color contrast to the rock.
Okay, what about neuromelanin? Is that any use?
Well, there we go. That's a question. Something to look out for. I'm not sure to what extent it has been done. And the problem there, of course, is that it will be present, if you think about something like a brain, it's a nicely sort of three-dimensional and rather decayable sort of structure, meaning that the matrix on which your melanocytes are sitting is going to kind of collapse on you, and this is, or in most instances it is, and this is likely to cause a problem. But it is most certainly something to look out for. I'm not sure that, I'm not totally up to speed in that area. It may be that the data have been published which I'm not aware of. If we think about the preservation of nervous systems, it's another of those things where synchrotron scans are probably going to be relevant. There's a sort of understanding, or there's an established but false idea among vertebrate paleos that soft tissue preservation is extremely rare. Now, part of the deal here is that if you start your study of the fossil by removing the rock to free the bones from the rock, you will probably also remove whatever traces of the soft tissues were left, right? And then you look at your bones and say, well, there weren't any soft tissues, because you probably didn't really see them. With synchrotron scanning, of course, we're looking at the bones often, in the rock. We have a much better possibility to see this kind of thing. And one of the key early discoveries from the application of synchrotron scans to early vertebrates was a beautifully preserved, probably phosphatized 3D brain inside the skull of an early cartilaginous fish of Carboniferous age. And it's lovely. You got the anatomical features, everything is Pradel et al., I forget which year, early 2000s. Very nicely preserved. There will no doubt be more examples of that kind of thing. We haven't found another one yet, but it'll come, I'm sure. Now that we're using a technique that is actually well suited to retrieving such stuff. And neuromelanin is certainly something to look out for. Thanks.
Per, you mentioned dentine found in teeth, but also in these scales of armored fish. So what came first, teeth or the scales?
All right, slightly complex question, that. What we do know is that among many early vertebrates, we have dentine either in the form of like separate little dentine scales or bigger bony plates with dentine ridges on them and stuff all over the body surface of the animal. We also sometimes have denticles in the pharynx. But immediately, if you're thinking about this from a sort of fossil, just studying the fossil perspective, you will realize that while it's easy enough to determine whether there are dentine scales on the outside of the body, actually finding what's inside the pharynx isn't so easily done. And so there's a reason to suspect, I think, that pharyngeal denticles are, as it were, somewhat underrepresented or understudied relative to the stuff that's on the outside of the body. It's evident that both systems go far back in time to before the origin of jaws proper. So teeth, teeth sensu stricto, are clearly a novelty associated with the origin of jaws. And when we find pharyngeal denticles of some sort in jawless vertebrates, nevertheless, they're always morphologically quite different from surface scales. Of course, we can't actually get at the molecular basis for the patterning here, but we do know from extant vertebrates that there's reason to believe that while you always have an epithelium and an underlying mesenchyme, that's where you're going to make teeth or tooth-like structures from. The key question seems to be whether there is an endodermal contribution to the epithelium or not. If you don't have endoderm, you're going to get skin denticles. If you have either all endoderm or at least some endodermal input, you can make teeth. Presumably, this is something that goes all the way back, and certainly among the forms that we're studying at the moment, which are some of the earliest toothed vertebrates, the teeth are always clearly distinct from the dermal odontoids, even when they're absolutely adjacent to each other. There's a crisp patterning boundary in there, even among very early vertebrates, which probably corresponds to the maximum extent of the endoderm, although we can't, of course, demonstrate that.
Hi. So, at the present, we have only two types of jawless vertebrates, the lampreys and hagfish, whereas their sister group, the jawed vertebrates, there are 60,000, 70,000 species. So how did they become so successful, whereas jawless vertebrates fell by the way? Of course, one can argue that jawed vertebrates were more powerful, ferocious predators. But there are also armored jawless vertebrates, which were probably as sturdy as those jawed vertebrates. So what are your thoughts on that?
Yeah, it's an interesting question. I mean, one thing to say, of course, if we look at the extant jawless vertebrates to start with, we know that lampreys and hagfish are sister groups. That's now well established on the basis of molecular data. We also know from the fossil record that certainly, by the Devonian, you have lampreys that look like perfectly good lampreys, and you presumably also have hagfish, although the record doesn't go quite that far back. both groups, although low in diversity, are evidently successful in their own terms. You know, they're quite abundant. They seem to have cornered their little bits of the market, as it were, and done that long ago and just stuck with it. But evidently, you know, jawed vertebrates have been able to radiate, as you say, in a quite different manner. And I would have thought that the key components here are the presence of jaws in the first place, but also I would probably point to having two sets of paired appendages. You get a quite different sort of maneuverability. If you look at the jawless armored vertebrates, some of the biggest ones were like kind of, they seem to have been filter feeding types with a width across the corners of the head shield of about, respectable animals. But you have either no pectoral fins at all or just one pair, and you had a kind of letterbox-like mouth, sort of a bit like a kind of manta ray, except that it's all you can do with it. And I suspect that these are major limiting factors. I mean, of course, there are other differences as well. We've got the extra semicircular canal in our inner ear, so we probably got better three-dimensional abilities to orient ourselves and so forth. But for my money, I would say two sets of paired appendages and the movable jaw. Perhaps one other thing as well. There's a bit more of a kind of odd subtlety. When you look at those early jawless fishes, back end of the body is covered in scales, front end is covered in big bony plates. We tend to refer to that as the head shield, but it's not actually a head shield, it's a head and shoulder shield. The front end of it, of course, is the front of the snout, but the back edge of that head shield is here. It essentially corresponds to our shoulder girdle region, and if you have pectoral fins in one of these things, like in osteostracans, they stick out straight out of the back of this head shield. What you have in jawed vertebrates, there's a patterning question in here that's really interesting and I think is worth exploring. If you look at the dermal skeleton of any jawed vertebrate, and I mean a modern-day bony fish would be a nice thing to look at here, but it works in terms of the morphology equally well with placoderms. Nice thing is, of course, the modern bony fishes is you'd have the molecular data. Look, the patterning of the dermal skeleton reflects and respects the underlying pharyngeal arch architecture. We have one set of dermal bones that belong with the dorsal part of the mandibular arch, another with the ventral part of the mandibular arch. If you have a gill cover, it's in the hyoid arch, etc. Right? And as part of that, you also have an occipital plane. So there's a separation between head and shoulder. In these jawless fishes, you don't have that. And the pattern of the dermoskeleton does not reflect at all the visceral arch organization, except in the kind of rather minimal way that there's a hole for the mouth and there's holes for the gills, and how would it work otherwise, sort of thing. But the point is, it doesn't allow for the same kind of flexibility of the front end of the animal at all. You have no ability to turn the head relative to the shoulder girdle. What you can do in terms of getting a vigorous branchial pumping function is probably more restricted. And things like this, I suspect, are going to be of importance as well. All of this taken together, I think, just gave the early jawed vertebrates a decisive edge in terms of ecological and functional flexibility.
Can I ask one more question? Yeah, even about the pectoral fin, the paired appendages.
Yeah.
So did the pectoral fin appear and then disappear in some lineages and then came back again along with the pelvic appendages?
It's a very good question, actually. The distribution of the pectoral fins among the jawless members of the vertebrate stem, of the jawed vertebrate stem, things like thelodonts and osteostracans, the distribution is patchy. It's uncertain whether pectoral fins are originating several times or whether they're originating and being lost again. But suffice it to say that they kind of, the sort of Schrödinger's appendages, they kind of flicker in and out of existence a bit in that part of the tree. Once we get into the jawed vertebrates, not only do you have pelvic fins for the first time, but the pectoral appendage remains constantly present, except in the sort of obvious exceptions like snakes. Yeah, so we get a locked-in arrangement of two sets of paired appendages. And of course, when you think about the pelvic fins, initially it looks like they don't do terribly much, perhaps. They're smaller and somehow less impressive than the pectoral fins. But they must be I think, functionally important right from the beginning, because they persist, and they would appear to have an important modulatory role of movement, even in something like a shark body, which in this instance I think is reasonably representative. It's a primitive pattern for jawed vertebrates. This is perhaps something that would be interesting to examine functionally. Then, of course, there's another matter that once you get to tetrapods, you get a switch where suddenly the hind limbs become more important than the forelimbs. We see this even in the earliest tetrapods. Suddenly the hind limb is bigger than the forelimb, whereas if you look at Panderichthys or Tiktaalik, pectoral fin bigger than the pelvic fin. And this kind of bigger hind limbs remains the case right up. We're absolutely typical in this regard, with a bigger and in locomotory terms more important hind limb.
I might have remembered wrong, so correct me if I'm wrong. I remember that in one of the slides you mentioned that there's a major milestone in the evolution of the fishes, that the inorganic component of their scale or their bone changed from calcium carbonate to calcium phosphate.
No. Our skeletons, the mineral component is always calcium phosphate.
Okay.
I think what you got mixed up with is that when you wanted to study fossils of these earliest vertebrates, they're often like these millimeter-sized dispersed scales in marine limestone. So how do you get at them? Well, here's the trick, because limestone consists of calcium carbonate. These little bits of bones consist of calcium phosphate, and that means that if you put your block of limestone in 10% acetic acid, it dissolves the calcium carbonate in preference to the calcium phosphate. So you get this residue at the bottom of the pot after all the limestone's gone, containing all these little scales and stuff. It's a very efficient way of collecting micro remains of vertebrates. There are, of course, other groups. If you think about the echinoderms, like sea urchins and stuff, which are rather closely related to vertebrates. I never showed them on the tree, but you had urochordates and cephalochordates, and the next branch out would be echinoderms plus hemichordates. Echinoderms also have a nice hard skeleton, which, like ours, lies within the body. It's not external, but it's made out of calcium carbonate, like limestone. And if you have a block of limestone with bits of, shall we say, sea urchin in it, you put it in acetic acid, you're left with nothing at all. The rock and the ossicles from the sea urchins just dissolve together. But because of this chemical difference between rock and bone, virtually all the work we're doing on dispersed micro remains, so isolated scales and stuff of early vertebrates, that work is being done using bulk sampling of limestone, which you then dissolve. And there's a point to that beyond the fact that sometimes that's all you can find. Articulated fossils of early vertebrates, a nice complete fish on a slab. This is really uncommon. You need special environmental conditions for the preservation. It's not necessarily going to be the case that this is representative of what's going on in the world at the time. But isolated scales that have just been like rolling around in the seafloor and eventually buried in the mud, these are easily preserved. They're common. It's rather like with land plants, the difference between finding entire land plants and finding the dispersed spores, which is much more likely. So what that means is that although the picture that you get from these dispersed micro remains is much more incomplete than, of course, having the whole fish, in some ways it's probably much more truthful. And when we can look at the marine record of these little bony bits and bobs, and we can say that you can trace now, you dissolve a nice bit of marine limestone that's anywhere down to like 500 million years old, and you're going to find these bits. And in older blocks, you are not. Suddenly there's no bone at all, no dentine at all. That very probably tells us that that's about the time frame when this stuff really originates. It's not very likely that we're missing a long sort of hidden prehistory of bone and dentine, because this is a sampling of kind of abundant fossils.
Thank you.
Thank you.
Detlev. I was absolutely struck by what you said about sponges and that they already have the genes for the presynaptic and postsynaptic proteins, right? Is there already some diversification of the cells, or do the same cells express pre- and postsynaptic proteins? Is there some form of a direction of neural transmission?
Well, this is ongoing research, in that we are just doing that in the moment. But it seems that it's the same cells, so that both the same cells have both pre- and postsynaptic genes. So that it is a capacity that they can both send the signal and answer, and then respond by contraction probably. So this is also physiologically, this is pretty well shown that this can go through the epithelium, and the cells both send and answer, respond to the signals. This is the research that Michael Nickel has done. Mm-hmm.
Have you done calcium imaging in sponges?
No, this has not yet been done. Yeah, the movie that I showed for Hydra is actually the first one for one of these basal metazoan nervous systems that really opens a new world there. Mm-hmm.
And how many cells express these presynaptic? What's the percentage of cells that express?
Well, it depends on what. I mean, they are scattered over all the cell types. Yeah, it's not that it is specific cells that express them all. So most of the cells, or most of the pre- and postsynaptic proteins, are still everywhere. The division of labor appears to be there to some extent, but only for a smaller set of genes. Yeah, which makes sense, because the evolution of cell types is about the division of labor, and division of labor in evolution progressing becomes stronger and stronger.
A very uneducated question. In both your areas of work, have you come across organisms that don't quite fit the story that you just told us? How do you deal with those, and other examples?
Well, if I may say something, so for example, the position of the ctenophores that I mentioned briefly in my lecture is of course exciting, in that they have a complex nervous system with partial centralization. They also have nerve net, but of course if ctenophores, which is still possible, populate the base of the metazoans, it would at least say that almost all components of the nervous system evolved convergently. Yeah, there might still have been some basic functions. I mean, as we know, we even find some of this in sponges, but then a large part of this would have evolved convergently. Yeah, and so there is still a lot of room where theories and hypotheses have to be adapted, changed. This is a very plastic and malleable area in the moment.
Exactly. That's a really, really interesting current. If I can just sort of flip that back to you, I get the feeling that your preference is very much to have ctenophores above sponges. I mean, if you get to choose which would be your favored phylogeny, just on the basis of, how things sort of,
Well, I would tend to agree with this most recent paper, as I think that in general, derivedness of groups, if they have a long time of evolution on a single branch where they just depart from everything else, this often leads to the effect that you have in phylogeny then this basal attraction or long branch attraction with the outgroup. And we are not working on this ourselves, so I can really just rephrase what other people say. But I would still think that sponges are most basal. Yeah.
We have, in fact, a really corresponding examples on the vertebrates. And of course, the short answer to your question is when we discover things like this, we ruthlessly suppress them and kind of gloss over it. But seriously, I mean, in fact, I glossed over one in my talk, which is that there's been a lot of debate over the years about lampreys and hagfishes. You see, it's easy enough to see, just you look at these things. The lamprey's got it's got a face, it's got proper eyes, it looks reasonably sensible. Hagfishes just look weird and kind of unpleasant. Hagfish, most morphologists, not all I should say, but most morphologists have concluded that lampreys are closer to jawed vertebrates than hagfishes are, so that extant jawless vertebrates would be paraphyletic. The molecular evidence, however, is overwhelmingly, consistently in favor of cyclostome monophyly, and in fact, of course, one can approach this in different ways, and there's a certain tendency to turn it into a kind of Punch and Judy theatre with the molecular and morphological data sets just sort of being pitted against each other in a sort of, oh yes it is, oh no it isn't sort of way, which isn't very helpful. What can be done, however, and I think this is a really exemplary piece of science. I have a colleague at the Center for Developmental Biology in Kobe in Japan, Shigeru Kuratani, who is really a developmental biologist with a particular interest in cyclostomes. His research group, they're the only people who managed to obtain hagfish embryos over the last century. Last time he worked was for a guy called Bashford Dean around 1900. Well, anyhow, what Shigeru and his people have done is look at the comparative development of these two groups and also go back to the morphological characteristics, look at hagfish properly and say, well, look, are they really as different as people have thought? And the answer turns out to be no. So I can take one classic example. It has been said that whereas lampreys, like ourselves, have blood that runs entirely in a system of blood vessels, it has been claimed that hagfish have a partly open circulatory system. In other words, suggesting that the blood partly just sort of oozes through the tissues, which is what you would find in Amphioxus. And of course, if this were true, it would be a major signal that those hagfish might be basal to the rest of us. In fact, however, this turns out not to be the case. What you do have in hagfish are great big subcutaneous blood sinuses, but they're lined with vascular endothelium, so they're basically just big, baggy blood vessels. You have to look at it carefully to see these details, but once you get there, you find, well, they're not as peculiar as all that. So actually, that story is straightening out. It's another matter when you have the kind of small-scale mess that I flagged up with Panderichthys and Tiktaalik. Or again, if we look among the earliest tetrapods, it's perfectly clear that these things are higher up in the tree than Panderichthys and Tiktaalik and below post-Devonian, kind of more modern tetrapods. But their internal relationships are a terrible mess, very difficult to resolve. It's simply because they're sort of small-scale parallelism. It's hard to say exactly who's paralleling whom, but it's going on. That's evident. In a sense, that is probably unresolvable. Of course, you can feed all this data into phylogenetic analysis and press the button, and a tree comes out. A tree always comes out if you press the button. It works every time. The question is how much faith you can put in the tree. But then again, maybe it doesn't really matter desperately. What is interesting in that kind of scenario, rather than get hung up on pulling out the correct tree here, is to perhaps recognize that the fact that we have areas of heavy convergence within the tree and then other areas where that's apparently not happening, so that the signal is much clearer, that is, that's data too. That in itself is saying something interesting about how evolution was working. In this instance, it would appear to suggest that right at the water's edge, there is, as it were, quite a lot of perhaps ecological experimentation and diversification with different things, trying out different degrees of terrestriality. And that's an interesting story, whether or not we can then fully resolve the tree that is produced. So that would be my take on it.
I have a question to Detlev. I was very much fascinated by your explanation of the development of synapses. Now, we know that in the human brain, the same neurotransmitter like dopamine can work in a non-synaptic or volumetric transmission way, or a synaptic way, and there is a terrible, significant concentration difference needed to obtain the same effect in a non-synaptic manner. So it's micro or nanomolar concentration, whereas in the synapse, the same effect can be achieved by femtomolar or picofemtomolar concentration. So it's a fantastic development for many reasons. But you showed these spongi, which show very clear movements. That is to say, their musculature or sort of musculature could have been driven by volumetric or non-synaptic neurotransmitters. And then when you have the synapse, the very same type of movement can be made by way of synaptic transmission. So have you or anyone else measured the concentration of neurotransmitters which achieve this sort of movement-like, or whatever it is called, movements in the spongy, and concentrations of the same neurotransmitter in animals which have already synapses?
Well, the answer is short. This has not been done, in that it is also difficult to do. What you can do is you can apply these substances and see the effect. But it's certainly something, and I agree that this is a fascinating question that should be done. And it is indeed probably just a gradual difference between the two. We see also the exciting question is how often has this gradual difference been walked through? So how often did it happen? Was it really only one event? Because we see a lot of situations where the situation is ambiguous between, I would say, clear synaptic transmission and a transmission situation which is, as you say, then rather volumetric and sometimes even hard to distinguish if you see EM pictures what this now exactly is. So the clear synapses, fast transmission, morphologically very complex structures are probably bilateral specific, and even there the question is, do we see convergencies? Yeah, so we are advocating, I think, that synapses, to some extent at least, might be homologous, for example, between the nerve nets of these lower metazoans, cnidarians, for example. But maybe this was still a rather primitive state and rather slow. Yeah, we have, by molecular comparisons, one can see that simply by presence in the genome, most of the components of cholinergic and aminergic fast transmission are bilateria specific. Yeah, then there is fast peptidergic transmission in cnidarians, yeah, which means they have established themselves a faster and more targeted transmission mode. And I think here the combination of very thorough analysis of the molecular components, it really matters whether they are the exact paralogs or whether they are some precursor that is still not having this precise function, in combination with localizing these into cells, and in combination with localizing into the exact cellular modules. Yeah, we will still see a lot of surprises, I think, in how the synapses really came together. And there are for sure many variants and gradual complexifications of a structure that is certainly the most fascinating.
The similarity of expression patterns of genes important for neural patterning was striking, right? Genes that control this in vertebrates, you find a similar pattern also in cnidarians and the other models you analyze, right? I was wondering about the neural crest. Are there genes that are important for neural crest in formation in vertebrates that you find expressed in your models?
The region where the neural crest emerges is old. This is exactly this boundary region between distalis and PAX-D and MSX that you even find in cnidarians. So this is an old, probably sensory part of the nerve net, yeah, that is also seems to be at least GABAergic to some extent. What is striking with the neural crest, I didn't speak about that, but if we look into the annulate and just go through the collection of markers that people know are specific for the neural crest invertebrates. So if we do the same thing as Marian Bronner, for example, has done for finding neural crest in lamprey, and then people looked at Amphioxus, the collection of genes that are expressed at the margin of the neuroectoderm in an annulate is much more complete than the one in Amphioxus, for example. So there is it's clearly an old region that has produced several cell types that still the neural crest produces, that has a certain specifics, very peculiar features that made it so important in the vertebrates.
But they are not migratory.
No, but they undergo epithelial mesenchymal transitions and so also move. So what is probably one of the answers to understand what is really special about the vertebrate neural crest is to look into what do these cells do during their development, what is not found in, for example, in an annulate, where you have pigment cells, where you have peripheral sensory neurons of the exact same kinds with the same molecular address and so on. So what is not there is that there is the closure of a neural tube, which is a fascinating evolutionary event in that you must have had a loosening and a resealing of epithelia. You know, there were folds coming, and then the end result is that they come together differently. This is in development, this is not a problem. In evolution, it is amazing. So there is a sealing of folds. And this is where the neural crest has emerged, probably out of a permanent adult mesenchymal situation, that these cells, and probably this conveyed them some potential, that they became this developmental joker, that they could go everywhere and take over the even development of bones, yeah, and even to more and more extent. So this is distinct in neural crests and invertebrates.
Yeah, I wanted to make a little comment or observation. It just relates to something I sometimes take up with my teaching with the undergraduates, and sort of plugs into the theme of your talk as a whole, in that it struck me what peculiar organisms animals are in the kind of, you know, the organismal world generally, something that's perhaps not always fully appreciated. Point is this. If we think about single-celled organisms, we know, I mean, obviously some of them just kind of sit around, but there are quite a few, and of course, very traditionally, they have often been viewed as animals, at least in kind of inverted commas, thinking now about, paramecium, creatures like this, that are able to do things quickly. What I'm getting at is that they're very reactive, and they can move, a good few body lengths in a second, this sort of thing. This works fine, potentially, if you're a single-celled organism. Indeed, I'm thinking again here about, your nice animation of the one conoflagellate swimming along early on. But typically, of course, if you start building a multicellular organism, you lose the ability to do that, because all these individual cells sitting together, because in a sense, almost as it were, of statistical or kind of cloud effects, you lose that ability. And so perhaps a rather typical multicellular organism is something like a mushroom or a tree. Not unresponsive, of course, but responding on glacially slow timescales by comparison. What is so bizarre about animals is that because of evolving this kind of couplet of a nervous system and muscles, we have uniquely been able to translate that level of scale, that level of motility up into kind of macroscopic multicellular scale, and which is what makes us so very distinctive in this world. So.
Thank you, Per, for this remark, because it's exactly right. I mean, early animals were actually not small, and if you move with contraction waves or change your shape with contraction waves, you're sort of amoeboid, but you're not really progressing in the same way as unicellular organism can. And I think this movie that I showed with the nerve net, almost simultaneously lightening up in the entire body length of Hydra, this very much looks like exactly this function. Yes. So that it evolved and what it made possible this way was to contract an entire body in a whole side, to maybe steer, to maybe retract the entire body, so to reinstall this function. That it's exactly like that was missing in early animals, and that other multicellular branches also didn't do. Yes.
Any further question? There's one here. Do we have a microphone?
You finish my question. I think I can rephrase it. It's a very general question, because, for example, in the shells and also some squirrels, when there is a need to build part of the body to be stiff to support the body, they didn't choose to use calcium phosphate. Sometimes it's calcium carbonate. And I have this question because these shells, crabs, squirrels, and vertebrates, they are all original from the ocean. So I'm wondering why there's such a difference that different animals choose to use different inorganic feeders to build their bone. And there must be an advantage or disadvantage, or there's or let's say maybe they choose what is more adaptable. Then, if that is the case, what is the pressure behind that?
That's a really good question. I say immediately that I don't have a good answer to it, but I think it's an excellent question and something that wants investigating. And I think I can tell you which is the group that might provide a clue. There's one group of shelled organisms that will flip between these two, and it's brachiopods. These superficially rather clam-like creatures, which are actually lophophorates related to bryozoans and things like that, and first appear in the Cambrian. There are brachiopods with calcium carbonate shells, and there are brachiopods with phosphate shells, calcium phosphate shells. There is a phylogenetic pattern to it. They don't just kind of flip freely back and forth, but nevertheless, rather closely related groups of brachiopods have opted for the one or the other. But what the, as it were, physiological basis for choosing one over the other, what that would be is very difficult to pin down. Vertebrates, I mean, some tetrapods use calcium carbonate in eggshells, but our skeletal tissues, all of them, bone, dentine, calcified cartilage, enamel, if you have it, it's always hydroxyapatite that is used. It's presumably because it's utilizing the same molecular pathways in the secretory cells, and you're then just kind of deploying the stuff differently. But I mean, yes, it's likely that there are selective factors here, both in terms of mechanical properties and perhaps in terms of it relating to the availability of particular ions in the environment that you inhabit. But I really don't have an answer to that, except to say that it's a very good question and will be interesting to pursue. So that's a non-answer for you, but thanks all the same.
I just want to say, I asked this question because I study materials, and
right,
like synthetic bone substitute, and we know that depending on the chemistry of your implant, some of them will be dissolvable in your body, some not dissolvable, some resolvable, some not resolvable, and that I'm thinking that there might be a reason from the original bone.
yes,
Yeah, and
that may very well be the case. One thing that's important to appreciate, by the way, with bone in particular, is that it's a very dynamic sort of tissue. If you remember that synchrotron model I had of some placoderm bones early on, absolutely shot full of blood vessels. You have a huge contact area here between capillary and bone, and it's fairly obvious that the bone is being used probably to modulate calcium and phosphate levels in circulation in the organism. They may also be using some of those spaces for putting hematopoietic cells in or whatever. It's very hard to know with these early vertebrates, but it's an enormous difference compared to, let's say, cartilage, which is just this kind of basically inert tissue with almost no vascular supply. So it's likely that bone has always had a seriously physiological function, as well as being mechanically important for the animal. That would appear to go right back to its early history. And quite early on, although not right at the beginning of bone evolution, quite early on, starting really with the placoderms, we start seeing evidence for bone resorption and regrowth, often quite sort of dramatic remodeling happening. And again, it's evident from the way the resorption surfaces are deployed. This is, by the way, something that we're studying now intensely on the basis of synchrotron scans, because you can see the resorption surfaces in synchrotron scans. You can model them in 3 dimensions, which has never previously been possible. And you can see how they essentially follow blood vessels, right? So it's evident that osteoclasts or odontoclasts, if you're looking at that, the cells that break down the hard tissue, are brought in along the vessels and kind of chewed things away, and then you redeposit, whatever, all of which partly, of course, is to do simply with reshaping your bone, but probably is also to do with, at various times, needing to mobilize the bone material for use in, physiological use in the organism. Thank you.
Do we have more questions?
Has there always been a difference between motor and sensory neurons, and if not, when does that first occur?
So the question is the difference between motor neurons and sensory neurons.
Has there always been a difference between motor and sensory neurons?
No, this is also a very nice question, and the answer is that they probably started in one cell type. In that, I mean, in a way for sure, because both sensory receptors and also some of the signaling substances would be in one cell from the very beginning. So there must have been a division of labor of some neurons then specializing on the sensory part and some on the motor part, on the output part. Furthermore, there was then a specialization, probably still from the sensory side, into interneurons. So the real first neuron would have been the one that was really only associative. And this happened, or this is present in the level of the nerve net organization in cnidarians and also ctenophores, where you have neurons that are just neurons. Apparently, I mean, we don't know to what extent they might still sense chemosensory or something. And of course, we have it in the bilaterians. But I would like to stress that, and curiously, even in Amphioxus, there are the motor neurons that innervate or seemingly innervate the muscles, and that in fact the muscles get the innervation by contacting the basal lamina of the neural tube. These neurons are in fact rather interneuron-like and have some collaterals that then go to the basal lamina. And on the other hand, they still have a sensory apparatus, which is contacting the central spine, the fluid of the neural tube. So even there, at this level, there is still all in one cell. So probably it's one of the principles almost of nervous system evolution is a constant division of labor, distribution of sensory motor function into segregated cell types that were initially in one.
Right. Quick follow-up question on that. Do you think that the transmissions would then have gone in both directions in the same cell, or one direction in one cell and another direction in another?
The cnidarian nerve nets are full of bidirectional transmission, yeah, but very often in different little spots. Yeah, so they are bidirectional synapses as well, but in most cases, the neurites would be both directions, but it would be signaling would be in different directions in different spots.
Thank you.
Time flies, and we're running late. So, is there any last pressing question? If not, I really want to thank both speakers. Excellent talks, very lively discussion, and I thank you for coming. And please join me for a round of applause for our speakers.